I'm Hadas Benisty, and I'm a member of the Faculty of Medicine at the Technion. I'd like to thank the organizers for inviting me to speak of our work. In this talk, I'm going to present two papers dealing with completely different questions and hypotheses, and even different modalities. The thing they have in common, other than me as one of the authors, is that in both papers, we developed a formulation to analyze the dynamics of functional connectivity. We utilized this analysis in two timescales. In the first paper, we looked into instantaneous changes in functional connectivity of the entire cortical surface and its relation to behavioral metrics indicating alertness. In the second paper, which was published a couple of weeks ago, we looked at longer timescales of days, where we showed that during motor learning, the naive network reorganizes towards a specific expert configuration. And this process critically depends on dopaminergic axonal projections from the VTA to M1. So I've plotted here the outline of this talk as a trajectory in the state space of analysis and neuroscience. And we're going to start with a spontaneous behavior indicating alertness and its relation to cortex-wide functional connectivity. I will show our formulation that goes beyond linear modeling, but still provides interpretability. We will proceed with some graph theory and Riemannian geometry, and then we'll switch gears and see how we utilize this analysis to explore functional connectivity in the span of days during motor learning. Finally, we will get to this discontinuity, the circle, and see what we learned from more basic and linear analysis about the critical role of dopamine for inducing changes in the dynamics. So now let's start here where this arrow points. In this video, we see the dynamics of cortical activity recorded using wide-field calcium imaging by Sweta and Andrew at Mike Higley's lab. This mouse is head-fixed and able to run on a wheel. We monitored three self-initiated behaviors: whisking, running speed, and pupil size. These metrics indicate the state of the mouse and how alert or drowsy it is. In this early paper, we divided the data into segments of sustained states, 3 states of alertness. In the low state and in the intermediate state, the animal is not running. In the intermediate, the pupil is large and the animal is whisking, and in the highest state, the animal is running. We showed that there are significant differences in the activity patterns across these states of alertness that made sense. The more alert the animal, the higher the activity. This averaging is simple and effective, but it leads to a massive loss of data. We have no way of knowing what happens outside of these sustained states, and we can't say anything about dynamics. So we started with looking at time traces of the activity from small parcels, which are brain regions. And then we tried to see if we could relate them to the dynamics of these 3 behavioral metrics: pupil size, whisking, and locomotion. As we might expect, everything looks super correlated, and just using linear modeling should be enough to interpret the relation between imaging, which is here in the green x of t, and each of these behaviors, the blue b of t. But still, we wondered if we could learn more. So we took the Taylor's expansion of this function f. In blue, we can see the behavior signal. In green, the neuronal activity, which is the first order term. And in red, there is the second order term of the instantaneous interactions between brain region I and J, and they are evaluated based on a single time point. In practice, this is highly sensitive to noise. So instead, we evaluate the interactions over a short analysis window to obtain the sample covariance. This leads us to the final formulation of the model. This model uses a higher order and therefore goes beyond linear modeling and has the potential to yield higher accuracy compared to using just the first term. And we maintain interpretability, as we can evaluate the contribution of each of these 2 elements and relate them to biological entities, which are neuronal activity in green and the functional connectivity in red. This is an illustration of how we extracted these short-term correlations. We used the activity traces and a short analysis window to extract a correlation matrix. Sliding the window gave us a temporal series of matrices. On the right, I've plotted the temporal progression of some elements of these matrices, and we can see that these signals have their own dynamics. So we started the model with a high-dimensional signal of activity in green. Typically, from one hemisphere, we got about 40 parcels. So the green signal has 40 dimensions. Then adding the second term, we get about 800 pairwise correlations. So obviously, this makes modeling difficult, since we are limited in terms of the duration of the experiments. Still, looking at the traces, it is clear that there's some latent dynamics, so we don't really need 800 dimensions to capture it. Therefore, we wanted to extract a low-dimensional representation for this dynamics. And this brings us further in the trajectory to the point of discussing graph theory. The purpose of reducing the dimension is to extract a latent representation capturing the intrinsic structure of the signal, in this case, the low-dimensional signal that drives the dynamics of the correlations. In order to reveal these latents, we built a graph where each node represents the connectivity of the brain at a specific time point. We used a diffusion kernel to evaluate the similarity of different time points, and then we extracted the latent dynamics of the correlation signals by taking the eigen decomposition of the normalized kernel. But I have left out how to evaluate the distances between the matrices, and this is actually not as simple as one might think. And this brings us further in the trajectory to the part of Riemannian geometry. So why not use Euclidean distances between correlation matrices? So I would say that potentially, given a lot of time points, many more than we had, it might have worked. But in the experimental setup of data extracted from 1 hour, we did not have enough data to overcome the fact that correlation matrices are not Euclidean entities. Fact is, they live on a curved manifold shaped like a cone. And this is because correlation matrices are symmetric and semi-positive definite, which means that their eigenvalues are larger or equal to 0. In order to correctly evaluate the distances between two matrices, we cannot use the Euclidean distance between them. We should evaluate the length of the arc on the manifold, and the way to do that is to use Riemannian geometry. And luckily, we do have a formulation for this distance on the cone, and we also have code for this. So I'm going to spare you a slide of equations, but please trust me that it does exist, and I'm super happy to answer any question about it afterwards. So to recap, we built this graph of matrices and computed pairwise Riemannian distances between correlation matrices related to different time points. Then we extract the embedded matrices to obtain a low-dimensional representation for the dynamics of functional connectivity. Which brings us back to the model. So we wanted to measure the contribution of each term. First, we trained how well we can predict the behavior using both activity and connectivity dynamics. And these are the blue bars, which you can see that are fairly high. For example, for predicting pupil, we get almost 0.6 R², which is fairly high. Then we shuffled the latents of the connectivity to see how well we can do without the connectivity dynamics. And these green bars are significantly lower than the full model. Then we shuffled the activity and saw that the accuracy we got, the red bars, was similar to what we got from the full model. So to conclude, using this analysis, we showed that fast changes in connectivity are significantly more related to changes in these spontaneous behaviors than activity. Now I'd like to present another context, in which it's interesting to look into connectivity dynamics, and that is learning. So I'm going to present the experiment first. It's going to be several slides before I get to the analysis. What is learning is a big question, and we see learning as a dynamic, adaptive process aimed at acquiring and optimizing performance. For this purpose, the brain receives new information and adapts by modifying its activity patterns. This is done through mechanisms of synaptic plasticity, and that is why looking into changes in functional connectivity is natural for the study of learning. Motor learning specifically is the process of acquiring skilled movements that require an association between sensory input and action planning. This is done through a highly distributed system with a primary motor cortex, M1, as one of its main hubs, generating motor commands for the downstream execution centers. Previous work shows that pyramidal neurons in layer 2-3 of M1 undergo plasticity changes during motor learning. And motor learning has been shown to induce structural changes in dendritic spines and axonal buttons. Still, it is unclear how these changes relate to changes in network dynamics. On the functional side, researchers use large-scale in vivo recordings in learning animals to explore changes in ensemble dynamics. But usually the effect of learning on functional connectivity is overlooked. Dopamine plays a central role in learning. It is a neuromodulator carrying information regarding outcome and reward. Thereby, it's driving plasticity at the synaptic level. The VTA is the main source for dopaminergic projections to M1. Taking these two facts together, we asked two questions. First, how does the pyramidal network reorganize during motor learning? And second, what is the effect of these projections on the network? Our working hypothesis for answering this question is that dopaminergic projections to M1 directly drive synaptic plasticity mechanisms, and this leads to changes in network connectivity, which in turn underlies motor learning. So to test this hypothesis, we designed an experiment in which we recorded the activity of the same population of pyramidal neurons in layer 2-3 of M1 during several sessions as the animals were trained to perform a hand-reach task. Amir, the student from Jackie Schiller's lab, trained these mice to reach and grab a food pellet once a go cue was heard. We had a second group of mice where Amir specifically blocked dopaminergic projections to M1 from VTA. All animals were trained till first success and started to record the neuronal activity. After the first session, we randomly assigned animals into groups. A manipulated group, which on training days 2 to 4, we injected CNO locally to M1 to inhibit the dopaminergic projections from VTA specifically. This group resumed training with no CNO on the 5th training day and continued training till the 7th day. Animals in the control group were trained for 7 days with no blockage. First, let me convince you that animals did learn the task. So what we see in this figure is a gradual improvement in execution in the success rate until reaching a steady level. Looking at the manipulated group, we see that injecting CNO did not prevent motor movement. It did prevent the steady improvement in performance till training without blockage resumed. And now, since synaptic changes drive learning, we wanted to see if we could identify a reorganization of the network in terms of functional connectivity. So in this setup, we had more structure than in the previous setup. Instead of a sliding window, we evaluated the connectivity on a given trial, and we did that for all the training sessions. And the aim of the analysis was to estimate if and how these matrices evolve with time. Then we used Riemannian geometry to do three things. First, to estimate the connectivity on a given training day as the centroid of the trials on each day. The second is to explore if we can identify that learning induced a monotonic reorganization toward an expert connectivity. And three, reduce the dimensions of the matrices so we could visualize how functional connectivity evolved with training. In this figure, we see the dimensionality reduced matrices of two animals along the training days. On the left figure, we see a control animal, where each dot represents a correlation matrix. Colors stand for training day. And we see that matrices are similar along trials of a given day, but progress and differ between training days. We can actually see a trajectory going from the blue, the first day, through the green and yellow towards the red. For the manipulated animal plotted on the right, we see that days 2 to 4 do not progress when CNO was applied. They seem like a big mix, and progression starts happening on the 5th day, which is the yellow day, and after lifting the CNO, meaning after training without blocking of dopamine. So to quantify this phenomenon, we used 3 strategies. The first one is a microscale analysis, and this was to measure the Riemannian similarity between the centroid matrices. Thus, we could account for the specific configuration of pairwise relations of neurons. The second is a mesoscale analysis, which takes the degree centrality of each centroid and measure how this evolves with training. And this analysis accounts for the average strength of connection each cell has to the network. And the third is a macroscale analysis, where we looked at the total average connectivity of the network. So first, I can already tell you that the overall average connectivity was constant, meaning that the overall value of the connectivity of the network stayed stable during learning. So we didn't get more connections overall or less. There was a change, but it was balanced. At the micro and meso levels, we plotted here the Riemannian and degree similarities between sessions as heat maps. So since we had 7 training days, you can see that each heat map is a 7 by 7 matrix. And the traces that you see is the values of these heat maps at the 7th day. So each trace is actually the similarity of each session to the 7th day. The control group shows a gradual progression towards the 7 days configuration. Injecting CNO delays this process. So overall, we see that during learning, the functional connectivity changes and progresses towards an expert connectivity configuration, and that process does not happen when blocking dopamine signals coming from the VTA to M1. And now I'd like to show you some results of simpler analysis that shed more light on the dynamics of neuronal activity during motor learning. So first, we asked whether we can see a transformation of the population dynamics in the activity as well. So up until in this stage, we showed the transformation in connectivity. So now we wanted to see if we could see the changes in activity. So specifically, we asked whether the ensemble activity of an expert animal is different from a beginner's, and if so, does learning causes the network to smoothly transform from one to the other? So to test this, we trained a model to separate between the ensemble activity of an expert animal from the activity of a beginner. And we then applied this model to all the training sessions. And then we counted the fraction of trials of each animal that was considered as expert. So for the control group, as learning progressed, more and more trials were identified as expert. However, for the manipulated group, during the CNO injection days, the activity was not consistently reorganized as either beginner or expert. And you can see that on days 2 to 4, we can see about 0.5. I mean, we can't tell if it's more beginner or an expert. Only when training with no CNO resumed that the ensemble dynamics became more similar to the expert dynamics, meaning starting from the 5th day of training. In our previous work, we showed that a small subpopulation of cells in layer 2-3 of M1 that reports the outcome of the trial. And these cells, we call them success-related neurons or failure-related neurons or outcome indicative neurons. And we could actually see, you can see here examples of several cells that are active only on success trials or only on failure trials. We also showed that this signaling was unrelated to movement, as it began well after the movement was over. So in this project of learning, we wanted to investigate whether this signaling is innate or does it emerge with learning, and how the dopaminergic signaling influence this emergence. So we applied a similar analysis to the one we did in the 2020 paper. And this is a very simple analysis where we trained a classifier to separate the activity, to use the activity of a specific cell to separate, to cluster whether a trial is success or failure. So if the activity if a cell is indicative for outcome, then we will be able to successfully train this model. So we did that for all the cells in all the animals and counted how many indicative cells we got per animal. And detecting these outcome cells on each training day in both the control and manipulated group. And we see that for the control group, on the first day, we didn't get that many indicative cells at all, but very quickly, this subpopulation emerged to the same, roughly the same fraction of cells as in our previous paper from 2020, about 13%. However, we see here that this process depends on dopamine reaching from the VTA because we saw no indicative cells during the CNO days, but they did evolve once training with no CNO resumed. Next, we explored how different variables of motor movements are represented in the activity of layer 2-3 neurons and if that changes in the course of learning. So we created a set of predictors based on the animal's motor movements and used them to train a generalized linear model for predicting the neuronal activity of each cell. So the set of predictors was the hand trajectories, which we extracted from the front camera and the side camera using deep lab cut. We also had orofacial components extracted using face map, meaning whisking. And we had binary temporal events such as lift, reach, grab, to mouth, at mouth, supinate. And since these binary temporal events don't have dynamics that matches the dynamics of calcium, we convolve them with a set of splines with different widths to match the dynamics of calcium. And also the signal of outcome we used as a constant signal with all ones for success and all zeros for failure. I did forget here to add the go cue, which we added to this model to see if there was also a representation for the sensory stimuli. What you see in this figure is the relative contribution of each component in the model for modeling the neuronal activity during the first day in training. So basically, we took all the predictors, trained a full model, and then we shuffled each component and retrained a partial model and measured what we've lost. And what we've lost, this is the contribution of this component. And we repeated that for every neuron and for every component and for every training day and for every animal. So what you see here is the aggregation of all these analyses. So a very, very simple linear analysis, which gives us a lot of work, but teaches us a lot, as you can see. So this is a relative contribution on the first day. And it shows that the first day, the activity is mostly correlated with the sensory signal, the go cue, and also to the outcome, but to a lesser degree, because this bar is slightly lower. As training progressed, and here I show the relative contribution on the third day, we see that the tone is less pronounced and the outcome becomes more significant. And this trend continues to the final day in training. So overall, we get a shift from the sensory input of the tone towards a representation of outcome. When we applied the same analysis to the manipulated group, we saw that blockage of dopamine from the VTA prevented this process, because we see that on the third day of training, we see that the bar for the tone is actually higher. But as training resumed without CNO, the final results state the outcome was the most significant component in the trained animal. So I guess I'm not used to not being asked questions during the lecture, so this is my almost final slide. Overall, in this talk, I've presented how geometric approaches applied to correlation matrices can help us understand the dynamics of functional connectivity. And specifically, I showed that fast fluctuations in connectivity are strongly related to spontaneous behaviors related to alertness. And then we demonstrated the critical role of dopamine signaling from the VTA to M1 in reorganization of the pyramidal network of layer 2-3 in M1 during motor learning. And I would like to thank my collaborators, Mike and Rafi at Yale, and Jackie and Ronen at the Technion. I'd also like to thank you for listening, and I'd be happy to answer questions as we have tons of time for that now.